evening to all the CUET aspirants. So here we are in our second session of embryology, where we are going to, till yesterday we have completed up to ovule structure and types of ovule. Till here it is done, right? So till yesterday ovule structure and types are done, and today we are going to exercise megasporogenesis, female gametophyte, that is embryo sac development, how embryo sac is developed, and then pollen morphology also is done. Pollination we are going to deal with, pollen pistil interaction and fertilization and double fertilization. These are the concepts that we are going to cover today, and post fertilization events we will look into the next class. So yesterday we are done till here, and now if we have to start with megasporogenesis, so here is the concept. What is megasporogenesis? Before getting into the megasporogenesis concept in detail, once let us look, just a second. Yeah. Once let us look into the female reproductive structure, okay, that is carpal unit. So yesterday we have learnt a single carpal unit will have stigma, style and ovary, right ma? So here if we have to see, yeah, stigma, style and ovary. Inside ovary there will be placenta and locules which are present. Inside locules you will have ovule. Fine, right? Ovules are present inside the locules of the ovary. And inside ovule we have learnt what are the labelings of the ovule diagram. Okay, what is the structure of ovule and what are different types of ovules based on different plants. And mostly in angiosperms it will be what type of ovule? The ovule is inverted. Fine, right? You will have inverted ovule like this. Fine, right? So inside ovule the complete cells which are present, these cells are known as nucellus. The cells which are present inside the ovule are nucellus, whose ploidy is diploid. Remember carefully, listen carefully. So ovule in most of the angiosperms it will be inverted, which is bitegmic, covered with two integuments. It has a funicle and a hilum which attaches the funicle, which is the stalk of the ovule, with the ovule body. Inside the ovule body you have nucellus cells. These nucellus cells ploidy is diploid. From here you listen the story carefully. One of the nucellus cell in the micropylar region gets differentiated into megaspore mother cell. Okay, from here the concept of megasporogenesis gets started. What is megasporogenesis? The production or the synthesis of megaspores which are haploid from a single functional megaspore mother cell which is diploid via one meiosis, and there is only one functional megaspore. That one functional megaspore from megaspore mother cell is formed via one meiotic division or reductional division and three sequential mitotic divisions, or you can say three sequential mitosis. Here if you observe, can you see in the diagram clearly? At the micropylar region, one of the cell is differentiated to megaspore mother cell, and this megaspore mother cell, after meiotic division, after meiosis 1 and meiosis 2, 4 megaspores are formed as such. Okay, 4 megaspores are formed. 1, 2, 3, 4. 4 megaspores are arranged in the form of linear tetrad. Okay, they are arranged in the form of linear tetrad, of which 3 megaspores towards the micropylar end will be degenerated and 1 megaspore towards the chalazial end, the base of the ovary. 1 megaspore towards the chalazial end will be functional megaspore. Okay, out of 4 megaspores which are formed in the linear tetrad, 3 megaspores will be degenerated and 1 megaspore towards the chalazial end will be functional megaspore, and this is haploid. Okay, so now from this one functional megaspore, 3 sequential mitotic divisions will happen to form the female gametophyte, that is embryo sac. I hope the story till functional megaspore is clear. Now we will begin with how an mature embryo sac is formed, how the development of female gametophyte is formed. Why embryo sac is considered as female gametophyte? Because embryo sac bears the female gamete, and the female gamete is egg cell. Okay, so one functional megaspore, if you can see here clearly, this one functional megaspore at chelysal end. Remember, megaspore mother cell is getting functional. Nucellus cell is differentiating into megaspore mother cell at which region? Micropylar region or micropylar end. But the functional megaspore is forming towards what? Chelysal end. Remember this carefully. This functional megaspore, after first mitotic divisions, after mitosis 1, it gets divided into 2 cells, 2 nuclei. Remember, every mitotic division is not followed by cytokinesis. Here karyokinesis is not followed by cytokinesis. So after first mitotic divisions, 2 nuclei are formed which are moved towards the extreme poles. One at the chelysal end and one moves towards the micropylar end. And second mitotic division, that is second karyokinesis, again two daughter cells which are formed are moving towards the either ends. And third mitotic division, you can see after third mitotic division, 4 nuclei are accumulated at each of the ends, 4 at the chelysal end and 4 at the micropylar end. Now after third mitotic division, after third karyokinesis, cytokinesis will take place. Cytokinesis takes place after third mitotic division. And here after cytokinesis, you can see 3 vegetative cells which are formed at the chelysal end are known as antipodals. These antipodals are also known as what, ma? Vegetative cells. Vegetative cells, and they get degenerated after fertilization. And then 3 cells at the micropylar end are addressed as egg apparatus. Okay, 3 cells at the micropylar end are known as egg apparatus. And here in the egg apparatus, what are the cells that are present? Synergids, and 2 synergids and 1 egg cell. These synergids are also known as cooperative cells or helper cells because they help in guiding the pollen tube towards the embryo sac. Okay, so synergid cells are also addressed as what, ma? Cooperative cells or helper cells. Cooperative cells or helper cells. Fine, right? Okay, these are also known as cooperative cells or helper cells, and 1 egg cell will be there, that is towards the micropylar end. One point to be highlighted again, here at the synergid cells there are filiform apparatus. What is this role of filiform apparatus? Filiform apparatus in the synergids, they will guide the entry of pollen tube, and one of the synergid, one filiform apparatus will be disrupted by the entry of pollen tube. When pollen tube enters into the embryo sac, one of the filiform apparatus will be disrupted. And at the center, if you see, 2 nuclei will together form 1 cell. At the center, this is known as central cell or polar nuclei. 2 polar nuclei at the center will form 1 central cell. But then all other cells present in the embryo sac are haploid. So the female gametophyte or embryo sac ploidy is haploid. Remember that. So once again I am telling you, from megaspore mother, okay, 1 megaspore mother cell at micropylar region, this undergoes reductional division, forming out 4 megaspores which are haploid. the megaspore which is towards the chaliceal end gets functional. 3 other megaspores which are towards the micropylar reach, and 1 functional megaspore. This functional megaspore which is haploid will be dividing. That is mitotic division only in the diploid cells in the animal cells, whereas in the plant cell both haploid and diploid cells can undergo mitosis. One standard example is the functional megaspore. Functional megaspore which is haploid can undergo mitotic division. It undergoes first mitosis, but here up to first to third mitotic divisions only karyokinesis takes place. Okay, only karyokinesis takes place, where one nuclei divides into two nuclei, one moves to micropylar end and one to chaliceal end. Second mitotic division, 2 nuclei on the poles. Third mitotic division, you can see 4 nuclei accumulated at each end. After third mitotic division, cytokinesis takes place, wherein 3 vegetative cells which are known as antipodal cells are formed towards the chaliceal end, and 3 cells are known as egg apparatus which are formed towards the micropylar end, of which 2 are synergid cells and they will be having filiform apparatus. One of the filiform apparatus will get disrupted with the entry of tube. Fine, right? And at the center you can see the central cell which has 2 polar nuclei. Fine, right? As all the cells in the embryo sac, ploidy is haploid, embryo sac ploidy is also haploid. Only exception with the polar nuclei. Till here everything is clear. And then look into the structure of embryo sac. Now you have clear embryo sac. So with the pollen. First I'll explain you the pollination as well, how it happens. So here if you see the antipodal cells which are vegetative cells, egg cell together called as egg apparatus at the central cell. So here whenever the pollen grain 2 volumes, right, they will land on stigma. Yeah, let us suppose this ovary is ovule, which has embryo. Egg cell over it, and at the center central cell. Fine, right? So here if you observe, pollen grain has 2 coats. Outer wall is exine, we know, and inner wall is intine. And pollen grains are non-motile. As they are non-motile, they are pollinated either through, they are taught to the stigma either through abiotic pollinating agents or biotic pollinating agents. Abiotic pollinating agents can be wind and water. Biotic pollinating agents can be any of the animals, birds, snails, snakes, and dominating biotic pollinating agent will be insects. So here when the compatible pollen grain lands on the stigma will receive the pollen grain. Stigma will be receptive to the pollen grain. How the stigma will be receptive to the pollen grain? Pollen grain will have S-locus cysteine rich proteins. Over the outer coat of pollen grain, S-locus cysteine rich proteins. These are known as SCR. Okay, these are the receptor proteins. And over stigma, if you can see, over stigma, there are kinases which are present. These are S-locus receptor kinases. Receptor kinases, very important, And at this chemical dialogue between pollen grain and stigma surface, calcium ions are richly present. Calcium ions, boric acid, okay, aquaporins, which lead to hydration and entry of the pollen tube over the stigma papillae. These are present over stigma papillae.
That's when a charged particle is present.
Fine. Apart with S-local cysteine proteins, on the outer coat of pollen grain, it also consists of lipids, which are triglycerides. Okay. Also consists of triacyl glycerides. So, after this huge chemical dialogue and the receptivity, from the germ pore regions over the outer coat, these are the small germ pore regions wherein there is no sporopollenin coat. From this germ pore region, pollen tube will start growing all across the style region. Clear, right? This happens after fertilization. And we know in most of the angiosperms, the pollen grain will be dispersed in two-celled condition, right? So, this has two generative cell. Generative cell will undergo one more mitotic division all across the style. Generative cell will undergo one more mitotic division, two male gametes. Okay, and the pollen tube enters into the embryo sac in any of the ways, either through the micropylar region, which is known as sporogamy, or through the known as chalcogamy, or through mesogamy. These are different entries of pollen tube into the embryo sac or into the ovule. Fine, right? So this will be the receptivity of pollen grain with surface. All the chemicals which are being presented here are very important, and be clear with SCRs and SRKs. SRKs are present over the stigma surface, SCR over the outer coat of the pollen grain. Okay, if you want, you can take a screenshot of this. And here, if I have to recall one more, there are different modes of embryo development. Like here, if you see, functional megaspore is developed into the complete embryo sac. This will not be the case with all the plants. Okay, so one functional megaspore developing into a complete embryo sac is addressed as monosporic development. Okay, this is monosporic development of embryo sac, wherein single functional megaspore the complete embryo sac. Fine, right? Two functional megaspores will lead to formation of a complete embryo sac. That mode of development is known as bisporic development. Bisporic development is allium, that is onion, and endymion. Okay, and that is known as tetrasporic development, where all the four megaspores are involved in the formation of the embryo sac. So that is known as tetrasporic development. Best example and go. Clear to here? Any doubts anywhere? Fine, right? Yeah. And then, as we have discussed about different, okay, so here we have two types of pollinations. One, flower, okay, in bisexual flowers especially, in bisexual flowers where both male and female reproductive structures are present, the pollen grains of the androecium of same flower are in receptivity of the stigma of the same flower, then it is known as self-pollination. Okay, in self-pollination also we have two types. One is autogamy, which takes place inside the same flower, and geitonogamy inside the same plant. Okay, means this plant will have two flowers, male and female flowers. It has male flower and female flower. Male flower and female flower are present on the same plant. Okay, so here flowers of the same plant pollinates. This will be genetically self-pollination only, but functionally it is cross-pollination. Geitonogamy is genetic and functionally fine. And then comes the concept of xenogamy. Xenogamy, two different flowers of two different plants. Pollination between two different plants. Here, self-pollination, as it is inside the same flower or in the same plant, this will not give you high variations across the generations, whereas cross-pollination will produce higher variations all across the generations as the pollination is in between two different plants. Fine, right? So, cross-pollination will help in increase of desirable variations across the generations. Self-pollinate will prevent that. So, to prevent self-pollination and promote cross-pollination, we have to learn out few concepts called as outbreeding devices. Clear till here? And then here you see from this diagram it will be clear to you. Autogamy in the same flower, geitonogamy of two flowers in the same plant, pollination in between two flowers of the same plant, xenogamy in between two different plants. And here prerequisites to occur. Okay, prerequisites for autogamy to occur. What autogamy takes place? It is very simple to be noted. Okay, autogamy occurs in bud pollination. Bud pollination see in pea and beans, where inside the bud, where it has male and female reproductive structures inside the bud itself, pollination happens in between them, and inside bud itself the pollination happens, thereby it will undergo fertilization. This happens in pea and bean. Clistogamous flowers. In flowers means the flowers which will not open at all. In few plants, if you observe viola or common pansy, oxalis and commelina, in these plants you can find both chasmogamous flowers and clistogamous flowers. Viola, common pansy and commelina, you can find out both chasmogamous flowers and flowers. Chasmogamous flowers are open flowers, whereas clistogamous flowers are closed flowers. These closed flowers will be present at the base, whereas chasmogamous flowers will be above. These flowers will not open at all. When the flower doesn't open at all, it doesn't have any other choice, so fertilization happens inside the same flower itself. So, clistogamous flower is one more prerequisite for autogamy. And third prerequisite is homogamy. What is homogamy? That maturity of both male and female reproductive structures in the same plant at the same time. When both male and female the same plant will mature at the same time, then there are 100% chances of self-pollination and autogamy. And in unisexual flowers, okay, a plant which is bisexual but it has unisexual flowers. For example, castor and maize, if you see. In castor and maize, you will have unisexual flowers, both male flower and female flower over the same plant. So, unisexual flowers in a bisexual plant, this will prevent autogamy, but this cannot prevent geitonogamy. Okay, so these are the examples and prerequisites of autogamy. Here and there we may get the statement based question, so then this is important. Fine. And then now we look on to the agents of pollination. We have both biotic and abiotic agents of pollination. Here we go with pollination, both biotic and abiotic agents. Abiotic agents, we are learning about both wind pollination and water pollination. Wind and water. Biotic pollinating agents, we have n number of biotic pollinating agents, but most important, dominated biotic pollinating agents is insects. First, here you see what are the characteristic features of a plant which is pollinated by wind. What are the characteristic features of this plant? Here you see a plant which is pollinated by wind. The pollen grains are non-sticky and light in weight. Okay, pollen grains are non-sticky and light in weight. Second most important feature is well exposed stamens. If you see male inflorescence, those are tassels. You can see best example is corn cob, okay, maize plant, wherein you can see well exposed stamens. Well exposed stamens. And if you can look into the tassels, that is female inflorescence, you can see feathery stigma, okay, feathery stigma. This is easily pollen grains, right? So well exposed stamens, feathery stigma, and pollen grains are light and non-sticky. And then each ovary will have single ovule mostly. Fine, right? So these are the characteristic features of the plants which are pollinated by wind. And if you can take the wide range of plants, they are pollinated by wind only mostly. Mostly they are pollinated by wind. Okay, and after this, water pollination. Water pollination. Pollination by water is again of two types. Firstly, water pollination occurs in just very few genera of angiosperms, like across 30 genera of monocots. Only 30 genera of monocots will undergo water pollination. Of this we have two types. We have epihydrophily and hypohydrophily. Epihydrophily and hypohydrophily. Fine, right? So epihydrophily, if you see, first, what are the examples of epihydrophily? We can take Vallisneria and Hydrilla. What are the characteristic features of the plants which are pollinated by water. Firstly, pollen grains should be light, non-sticky, and they should have a mucilaginous coat over it, followed by long pedicels if it is pollinated by epihydrophily, that is above water currents. Okay, then it should have long pedicels, and male flowers which are released into the water currents, they will travel across the water currents and they will reach the surface of the stigma which is above the water surface. Next is hypohydrophily. This happens inside the water in submerged plants. Best example you can take is Zoostera, that is seagrass. So here these plants, as they are underwater, pollen grains are ribbon like, so they can easily travel across the water currents. Few plants like water hyacinth and water lily, they are not pollinated by water, but then they are pollinated by wind. Clear, right? Clear till here? Any doubts anywhere, ma? And then?
Right.
Yeah. Pollination by insects known as entomophily, by bird ornithophily, by bat it is known as chiropterophily. These are different biotic agents of pollination. So here the pollen rewards that will get to the pollinators is mostly nectar. Okay. Nectar is the reward. Nectar, and few of them like Pronumba moth. Pronumba moth. What benefit does it get? It lays its eggs. Lays eggs inside the flower, inside the ovary. Okay. So this moth will misinterrupt thinking the flower as the male insect, and then it will lay its eggs inside the ovary. Okay. And wherein moth will get the benefit of eggs gets fertilized again inside the ovary only. By the time this turns to seed, the eggs will be dispersed. Fine, right? So this is the benefit that Pronumba moth gets when it pollinates yucca plant. Fine. So this is about insects, and the characteristic features of the plant, if you see, the flowers will be bigger in size, fragrant, okay, to attract the insects, okay, and they are of different colors, and pollen grains are sticky so that they can easily land over the pollinators and over the stigma. Okay, unlike the abiotic pollinating agents where they are non-sticky, pollen grains are non-sticky there, but here the pollen grains are sticky. And then outbreeding devices, as we have discussed. Outbreeding devices are the devices to escape the self-pollination and to promote the cross-pollination. Here we go. First is dichogamy, that is maturation of male and female reproductive structures in the same plant is at different times. If at all male reproductive structure is maturing fast, it is known as protandry. Okay, protandry. If female reproductive structure, that is gynoecium, is maturing first, that is known as protogyny. And heterostyly, when the length of style and the filament of male reproductive structures are of different lengths, which will resolve the problem of self-pollination. For example, tall male reproductive structures and short styles. Okay, here the self-pollination doesn't occur. Vice versa, long styles with short male reproductive structure, short filaments. Here also fertilization doesn't occur. And self-incompatibility, where the chemical dialogue that we have discussed, that is compatibility of chemicals between the pollen outer coat and stigma papillae, will not match in the same flower, that is self-incompatibility. Prepotency, hercogamy and dicliny are also the same. So these are the outbreeding devices. Clear? Here in this diagrams you can see protogyny and protandry, that early maturation, early release of pollen grains here in protandry, here stigma being receptive even before the pollen grain release. And here you can see the heterostyle with longer style and shorter filaments. Now, yeah, this concept we have learned, artificial fertilization or artificial hybridization. This concept is important, that in a bisexual flower, if we have two compatible parents of our desirable size, for example, male parent with tall and rigid stems, female parent with large fruits. From female parent, which is bisexual, the male anthers will be removed by the process of emasculation. Okay, and after emasculation they are bagged so that the outer pollen will not fertilize the female reproductive structure, post which pollination is done by our desired male gametes, and after which rebagging is done to prevent the pollution of other pollen grains. So here emasculation followed by bagging, pollination by desired male flower followed by rebagging will further you with the desirable characters of both the parents. This is artificial fertilization. Now we will learn the concept of double fertilization. How double fertilization happens. We have learnt about the pollen tube entry. It can be either through micropylar end or through chaliceal end or through integuments. It can be porogamy, chalicogamy or mesogamy. Here, once the pollen tube enters into the embryo sac, what happens is, as the inside the pollen tube only, what happens? Generative cell divides into two male gametes. One male gamete will directly fertilize with the egg cell, forming out a zygote, and another male gamete will fertilize the polar nuclei at the center, forming out a triploid endosperm. Okay. So here one male gamete fertilizes eggs. This forms out to be a diploid zygote. And second male gamete, which is haploid, will fertilize with polar nuclei, which is diploid, forming out a triploid primary endospermic nucleus. As here 3 nuclei are involved, this is also known as triple fusion. Okay, now let us have a glimpse on post-fertilization events. Post-fertilization events include zygote maturing to embryo, which is embryo development. Polar endospermic nucleus will develop into the endosperm, mature endosperm. So endosperm development. Here in most of the angiosperm, what happens is endosperm development will precede the embryo development. This is because endosperm will provide enough nourishment for the embryo to develop. Okay, and then seed formation, special types of reproduction, that is apomixis, and how multiple embryos are formed inside the ovule without fertilization, that is polyembryonic concept, followed by the objective related to reproductive biology, will be dealt tomorrow. So if we have to take out the key takeaways of today's class, what we have learnt? We have learnt about pollination, types of pollinating agents, abiotic and biotic, and then after pollen release, what is the chemical dialogue between the pollen grain and stigma receptivity with complete protein structures like SRKs over the outer wall or SCRs over the outer wall of the protein coat and SRK over the stigma papillae, followed by calcium ions, boric acid, and all we have learnt. And after which we have gone through the artificial hybridization, initially about megasporogenesis as well, and different types of embryo sac development, monosporic, bisporic and tetrasporic embryo sac development, and at last how the pollen tube enters the embryo sac by 3 different modes: porogamy, chalosogamy, mesogamy, with examples, followed by artificial hybridization and double fertilization. Here inside the embryo sac, whichever antipodals are present towards the chaliceal end and synergids and filiform apparatus near to the egg will degenerate after fertilization. Remember that. And always do remember the ploidies at each segment, because they will be asked along with the ploidy type of questions also will be asked with calculations of cell division. Okay. So, that is all for today's session, ma. In the next session, we will meet up with post fertilization events in plants.